Okay guys, so what this is going to be is just a review of what we went over in class today. And so for anyone who likes to hear it again, this is going to be the purpose, and I'm going to try and do it for every lecture from now on. So we are starting now chapter 13, and in this chapter what we're going to be looking at is the process of RNA and protein synthesis. And so the process in which RNA is made is called transcription, where proteins are made is translation. Okay, so transcription, translation, we're just going to be looking at RNA and the process of transcription today. So the drawing that I provided you with was really important because it shows a really nice analogy for the relationship between DNA, RNA, and proteins. So DNA, we've already been studying it. We studied DNA replication. We looked at that process of DNA polymerase and how DNA can be replicated. The whole point of DNA, though, guys, is that it's kind of permanent. You don't lose DNA. It's always, you copy it when your cells are dividing. But it's something like what we would consider a master plan. So when an architect is making up plans for a building, they make a master plan. And that is the final, the permanent copy that they have. And so that's what our DNA is. We don't change the amount of DNA we have. We always keep the same amount. We copy it if our cells are going to divide, but otherwise we want to just keep this master plan. Now, here comes the role of RNA. Now, if a builder is going to want to go and use his designs, obviously, to build a building, he doesn't want to bring his master plan because there's the possibility that, it could rain and it could damage it, and you don't want that possibility. Just like DNA, you don't want the possibility of having your DNA hurt. And so here comes the role of RNA. Now, if I take a copy of this master plan, what is created is what we call a blueprint. Now, a blueprint is a copy of the master plan that can get, rustled up, it can get dirt on it. We really don't care because we can always make another copy. It's super easy to do. So in this case, the master plan is the DNA, the blueprint is the RNA. So what RNA then does, because it's taken to the building site, RNA is responsible for the production of whatever they're making, or for us, what we care about, the production of proteins. So the order in which things go for the creation of your traits, what you look like, what causes that is proteins. Now, in order to create those traits, we've always said that DNA is responsible. Well, DNA goes and becomes RNA, and then RNA is what actually creates those proteins which make you and me the unique characters that we are. Okay? So DNA is considered the master plan, and the ultimate goal is to make proteins, but RNA is kind of that middleman in a sense. Okay? A couple of things for you to think about. When we talk about genes, okay, DNA makes up those genes. So it's a strand of DNA that has that is a gene. Okay, so a gene is made up of specific DNA that is responsible for certain proteins that are then made. So it goes, the genes, which are made up of DNA, become RNA, which then become the proteins, which are the traits, your hair color, your eye color, your skin color, etc., etc. So that's the order in which things are going to go. So let's really quickly compare DNA and RNA. Really quickly, what you need to know is that DNA and RNA are both nucleic acids. So they're very similar, except they have 3 differences, which we looked at in class. The 2 of the differences, guys, are really obvious in this drawing. One of them is not so much. So let's go over the 2 obvious ones right now. If you compare DNA and RNA, what you'll notice is that DNA has 2 strands. Okay? RNA does not. So that's the first difference. RNA is single-stranded and DNA is double-stranded. The second difference, if you look at the nitrogenous bases, you've got C, G, A, and T for DNA, and you've got C, G, A, and U for RNA. So that's another difference. There is uracil in RNA, there is thymine in DNA. Okay? And the last difference is not very obvious based on the drawing, but it is obvious based on the name of these 2 molecules. So here you've got deoxyribonucleic acid. Now, they're both nucleic acids, so they both have nucleic acid in their name. Notice the difference, deoxyribo. Deoxyribose is the sugar. Remember, you have a phosphate group, a sugar, and a nitrogenous base. Deoxyribose is the sugar for DNA. Ribose is the sugar for RNA. So that's a second difference, the fact that RNA has ribose for a sugar and DNA has deoxyribose for the sugar. Okay? Now, there are 3 different types of RNA that we'll be studying. We've got mRNA, we've got rRNA, and we've got tRNA. Okay? Now, these RNAs, and almost all RNA, are involved in making a protein. So we're going to look at how they are involved. So the first one's mRNA. Now the M stands for messenger RNA. And what these are, guys, this represents the blueprint from the DNA. So this was created from a process of copying the DNA, which we'll look at in a second. So mRNA actually has the instructions, the nucleotides, right, that then will enable a protein to be made. So this is the single strand. mRNA is the single strand that's created once it's copied from DNA. Okay. rRNA, which stands for ribosomal RNA, okay, makes up the ribosomes. Okay, so these there are two different subunits on a ribosome. You can see this first section, which is in blue, and then the yellow section. That's made up from rRNA and a number of different proteins. So that's all you need to know. rRNA makes up ribosomes. Ribosomal RNA makes sense. The last one is tRNA, which stands for transfer RNA, and that's right here. Okay, so this RNA molecule is responsible for transferring an amino acid, okay, to the ribosome. Okay, and we'll look at this when we look at translation. All you need to know right now is it's responsible for transferring an amino acid. Which amino acid is actually added depends on these three letters right here, and we'll look at that later on. So all you need to know right now, guys, mRNA is the long single strand of RNA that's copied from DNA, which we'll look at in a second. rRNA is responsible for making up ribosomes in conjunction with other proteins. So proteins and rRNA make up ribosomes. Okay, and tRNA is responsible for adding on amino acids one at a time to make a protein. Okay, so we'll actually mostly be looking at just mRNA today. We'll look at rRNA and tRNA in the next class. So this is a really nice just overview of everything. You've got your three different types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. If you notice, messenger RNA, just follow the flow chart, is also called mRNA, and it carries instructions. Okay, it carries those instructions from the DNA to the ribosome. Okay, and we'll be looking at this today specifically. rRNA, it's also called rRNA, and it combines with proteins to make ribosomes. And the last one, tRNA, works in order to bring amino acids to the ribosome. So these two we'll be looking at next time. All right, a couple more things. RNA synthesis. So this process of making RNA, guys, is called transcription. And the important thing for you to know, if you notice here, you can see the DNA right here being unwound. So just like in DNA replication, where we copied our DNA, the strands had to split apart, right? The hydrogen bonds had to be broken. That also occurs in RNA synthesis, which is also called transcription. Now, rather than having DNA polymerase go in and make a new strand, you have something that's called RNA polymerase. So RNA polymerase separates the two strands and then adds complementary base pairs for RNA to just one of the strands of DNA. So it's literally copying just the way that the DNA replication happened with DNA polymerase, but you're just adding one brand new strand. And once that's done, you have a brand new RNA strand, which is called actually mRNA. The important thing, guys, RNA polymerase adds base pairs that are complementary. Okay, so the new nucleotides that are being added are complementary. So if the DNA is A, the RNA would be U, right? Remember, uracil rather than thymine. If the DNA was C, the RNA that would be produced would be G. So it's just like DNA replication. Okay. A couple other important facts. In prokaryotes, this process occurs, but since they don't have a nucleus, it happens in the cytoplasm. And in eukaryotes, since they do have a nucleus, transcription happens in the nucleus so that we can keep the DNA safe in the nucleus. But then when the RNA is made, the new RNA, it's going to move out into the cytoplasm, and that's where proteins are going to be made. Okay. Couple of last things. I already talked about the fact that RNA polymerase separates the two strands, just like helicase did for DNA replication, and then it creates a complementary strand, right? So the RNA nucleotides are complementary, so if it's A, it's U, C, the RNA would be G, et cetera, and you end up just getting one new strand of RNA. Okay? The way that this actually works is really interesting. If you look at the DNA right here, okay, there is a section of base pairs, okay, and they tell the RNA polymerase to start. Okay, that's called the promoter region. Okay, so it's specific base pairs which signal the RNA polymerase to actually start making the new RNA. This is the equivalent of your RNA primer when we were talking about DNA replication. Okay, and so the RNA polymerase goes, keeps going, and then there's another region of bases that signal the RNA polymerase to stop copying. And so what ends up happening is it starts, it copies brand new RNA, the RNA polymerase goes, keeps going, and then stops. Okay? Once this is done, the DNA, which was unwound to make the copy of RNA, the single copy of RNA, it then winds up again. Okay? Now, here's the interesting thing about transcription. We've got this brand new strand of RNA. Okay? Before we go and take the mRNA and make it into proteins, we have to edit it. Okay? And so there are portions of this mRNA, okay, that are going to be cut out. And those sections that are cut out are actually called introns. Okay? So this is considered to be pre-mRNA, meaning it hasn't been edited yet. Okay? It leaves the nucleus, okay, and what happens is that the introns are taken out, they're spliced, they're literally taken out, and what's left are just these yellow regions. These yellow regions are called exons. Okay? So these exons are then put together, they're combined together, they're spliced together to make that final mRNA strands. Okay? Now, you might ask, why in the world is mRNA created in such a fashion that you need to get rid of these introns? And the honest answer is we don't know. We're not really sure why you have to get rid of the introns and then this is your final mRNA strand. What is interesting, though, is different regions can be spliced, can be cut out, and that leads to different proteins, and you'll see why later on in this chapter. So this process might have helped the whole process of evolution. So the fact that different proteins can be made based on where you cut out the introns. But it's just important to know that in order for the mRNA to be properly used in the cytoplasm outside of the nucleus, in order to make proteins, they first have to be edited. So you get rid of the introns and use the exons. So at this point, guys, we've talked about going from DNA to RNA. Once it's made into RNA, it's considered to be pre-mRNA because it hasn't been edited. Once it is edited, then it's in the cytoplasm and it can be used by ribosomes to make proteins, which is the next thing that we're going to study. And that's it, guys.